Going to get started today. So good morning, everybody. In case you don't know, my name is Reese Mazaid, and I work with Michelle Pardue. It's my pleasure today to introduce Dr. Marla Feller, who is a Paul Licht Distinguished Professor in Biological Sciences in the Department of Molecular and Cell Biology at UC Berkeley. Dr. Feller received her doctorate in physics, actually, with Dr. Yuan Ran Shen at Berkeley, and then proceeded to do some postdoctoral work with David Tank at Bell Laboratories, and then also some additional postdoctoral work with Dr. Carla Schatz at UC Berkeley as well, where she studied spontaneous waves of retinal activity during development. Before joining her current position at Berkeley, she also held some investigator roles at the NIH and also UC San Diego. Dr. Feller has contributed extensive service within Berkeley itself, but also to the visual neuroscience field and community, and participated in many of the conferences and committees within that community. Additionally, she also, I believe, has some editor roles at several journals, including eLife and Journal of Neuroscience. Dr. Feller also has a very impressive mentorship record with, I believe, somewhere around 40 pre- and postdoctoral trainees, which is great. And then it becomes no surprise that she's also been been awarded recently some teaching and mentorship awards. Additionally, she's also been recognized for her work. And for example, she was awarded the Brian Boycott Prize, which is a prize given to for achievements in your career in retinal neurobiology. And so her laboratory focuses mostly on investigating mechanisms of retinal circuit development and the role that retinal waves, the spontaneous activity, play in these processes. And as part of this work, she studies many classes of retinal neurons, including direction selective retinal ganglion cells, which we'll hear about plenty today. So with that, I'd like to give the stage to Dr. Feller.
All right, can people hear me? Is this, okay, great. So thank you all for coming. Reese, thank you for that very nice introduction. I've known Reese since he was a Ph.D. student working in a retina lab in Arizona. Thank you all for the invitation to talk to you today. And I'm going to talk to you about sort of one of the big questions, research questions in my lab, where we don't have the answer yet, but I think it's a good opportunity to sort of tell you how we're thinking about the question. And I look forward to having conversations with people to get all insights on what experiments they think that we can do in the future. And so, I'm going to spend quite a bit of time introducing this direction selectivity as a neural computation, because I think it's the intricacies of that circuit that we want to understand how it gets wired up during development. Okay, so and I'll tell you about the progress we've made along the way. Okay, so let me just start by introducing the retina. Let me see, is there a pointer? If not, can you guys see my arrow? I'll use this. Okay, so the retina is, we like to think of as a little piece of the brain that is external to what most of you probably think of as the brain. So light enters the retina, it passes through the neural tissue, it activates the photoreceptors that are at the back of the eye. The output of the retina are these cells that are called retinal ganglion cells. These are much more conventional neurons. They fire action potentials, and it's their axons that make up the optic nerve that carry all that information up to the brain. And in between those photoreceptors and ganglion cells are populations of excitatory and inhibitory neurons. And these are the ones that perform the neural computations like that of direction selectivity that we're going to talk about. So, a lot of people, particularly those who study visual cortex, like to think of the retina as just this very, very good camera, right? And if you look at the level of the photoreceptors, that is actually a pretty good assessment. So photoreceptors are very linear encoders of luminance across the visual field. There's only a couple different types. But when you get to the level of the output at the ganglion cells, there's estimated to be about 40 different types. And those 40 different types encode different features. By the time you get to the ganglion cells, you no longer have this luminance detector, but rather you have a bunch of feature detectors. And feature detectors are things like sensitive to edges, sensitive to color contrast, and sensitive to motion, which is what I'm going to be talking about today. And so in between these very few photoreceptors and these multiple types of ganglion cells are lots of interneurons. There are bipolar cells, which are feed-forward excitatory interneurons between the photoreceptors and ganglion cells, and a whole host of different kinds of amacrine cells, which are the inhibitory interneurons that also help shape these computations. Okay, so these are all this is schematics of the morphology, of the dendritic morphology of ganglion cells that are sensitive to motion. And there's multiple different types. They come in all sorts of sizes and all sorts of dendritic densities. They actually project different parts of the brain. And a subset of these motion sensitive ones, which are circled here, are sensitive to the direction that an object moves within the visual field, or the direction that the visual field itself moves. And that's going to be the focus of what we talk about here. So what does that mean? That means that here's our little cartoon schematic of a retina. And this is a recording that's being done from one of these direction selective ganglion cells. And if you take a bar of light and you move it across the photoreceptor, so you're sequentially activating those photoreceptors, you get a burst of action potentials at the leading edge and trailing edge of that bar. Take that same bar of light, you move it in the opposite direction across those very same photoreceptors, activating those very same interneurons in between, and you get very few action potentials, right? So something has happened between those photoreceptors and the ganglion cells that's broken the symmetry and has said that we're going to have a lot of action potentials sort of moving in one direction versus the other. Okay, so that's why and we think of this as a feature detector because it doesn't actually, this particular feature, which means a lot of firing and when the stimulus moves in the preferred direction and no firing when it moves in the other direction, is actually independent of the stimulus. So you can use drifting gratings, you can use a random dot kinetogram, you can use a bar of light. And in all cases, of course, the pattern of action potentials might be different, but the fact that there's no firing when that stimulus moves in the null direction is still true, right? And that's why we call it a feature detector, because the feature is the direction of motion that the stimulus moves and not anything else about the stimulus. Okay, so it's a great example of that. And I think I would like to say that this is not something that's unique to the retina. Direction selectivity has been found in many other neural circuits. There's examples in somatosensory system that whiskers moving in one direction versus the other might give you a different response property. Even for some place cells, if a rat is moving on a linear track, it moves in one direction versus the other, you might get different firing. So this idea that there is direction selectivity in stimulus space is something that's not unique to the visual system. Okay, so and in many ways it was considered one of the first computational neuroscience problems, right? When it was first proposed back in the 1800s as this idea, and how do we build a circuit that does this computation? And that's been it's been a long-standing problem. So we didn't pick this problem just because it's a cool thing in the retina. It's actually kind of a foundational question, I think, in neural circuits. Okay, so now let's go back to talking about it in the retina, where a lot of it is well understood. So even though we sense motion in all different directions, it turns out that these direction selective ganglion cells fall into different categories. And so if you look at, this is actually experiments that were done by recording from the optic nerve of a rabbit retina. And they found that if you look at the preferred directions of direction selective ganglion cells, they seem to fall into 7 categories. Sorry about that. So there are the on-off cells, which are the ones that I just talked about. They will give action potentials at the leading edge and trailing edge of the bar. So a dark object moving or light object moving will activate these cells. And they have preferred directions that cluster along 4 cardinal axes that we call them, right? So there'll be some direction selective ganglion cells that want up, some that want left, some that right, some that want down. There's another class of these, and these project to superior colliculus and to the lateral geniculate nucleus of the thalamus, and they're responsible for conscious visual perception of motion. The on-direction selective ganglion cells have slightly different preferred directions, and these are actually the cells that are responsible for a reflexive eye movement that's called the optokinetic reflex. So they project to a different part of the brainstem regions that mediate this reflex. And just give you an example of what this is the mouse direction selective, optokinetic reflex. So it's when the stimulus is moving across the whole visual field, your eyes will adjust to stabilize that image. So that's the reflex we have, that when we walk through the room, images are kind of stabilized because our eyes are reflexively moving to the entire visual field moving, as opposed to objects moving within the visual field. Okay, so those are all our kinds of direction selective ganglion cells that we have, and mice have the same complement as well. So it's been so some work that's been done recently. So back in the days when this was being studied in the rabbit retina, the visual stimuli were always given in the center of the visual field. But it turns out when experiments were done in mice and you actually moved to different parts of the visual field, you found that those preferred directions of the direction selective ganglion cells actually changed depending on where you were, and this and that they didn't actually encode, motion in the visual field, but they actually seem to encode motion of the animal. They followed an axis that was defined by what's called optic flow. So when we move through the world, right, objects are moving past our retina, and there's going to be a point where, because we're forward focusing, that as we move, images are stable. We kind of call that a singularity, but as you get farther and farther from the center, you'll get more motion on the retina as you move through the world. Now, mice, their eyes are kind of on the side, and so that singularity point is kind of located in temporal retina. It's kind of what's shown in this green dot here. And for ventral retina, if you move up and down, there's going to be a location that's in the ventral pole, and in the dorsal pole. And so it turns out if you look in the center of the retina, you'll see that the preferred directions are orthogonal to each other. But as you move, say, towards the ventral pole, you'll see that direction selective ganglion cells in this half are going to kind of point towards the center, and direction selective ganglion cells in this half are going to point towards the center. And so they kind of have this convergence, almost like, think of it like a globe, right? Like how we think about latitude and longitudinal lines on a globe, longitudinal lines. Longitude, right, where they point to the poles. So that's kind of what the map is like in mouse coordinates. And it's a very interesting idea, that it's kind of movement of the animal that dictates where at any given point on the retina, what direction that cell is going to encode. And this is work actually that was done by Shai Sabah, who's a postdoc in David Burson's lab. Okay, so we are interested in this map. We call that like a map of direction selectivity across the retina. And so the way that we are going to describe this map is by doing population calcium imaging. And we do that using a 2-photon microscope. So here's what we dissect the retina out of a mouse. This is what it looks like in the chamber. We put that chamber under a 16x objective that allows us to image from a large population of retinal ganglion cells, and we do calcium imaging to do that. And so we do 2-photon calcium imaging, and then we actually we've modified our microscope so that we have a visual stimulus that comes in and projects, say, drifting gratings. And so what you're looking at on the right is a field of view. The red squares, what our field of view is, and then these purple bars are the stimulus that are going to move through that field of view. And I should say these are experiments that were carried out by Alex Turiak while he was a postdoc in the lab. Alex actually just started his own lab at Vanderbilt University. And these experiments are also being done by Karina Bystron, who's a current graduate student in the lab. Okay, and this is now what their data looks like. So the calcium dye we use is called Cal 590. What you're looking at here are, these are the ganglion cells. These large ribbons are the blood vessels that are on the surface of the retina. And hopefully you'll be able to see as a visual stimulus moves through that we get this really beautiful activation of lots of ganglion cells, most of the ganglion cells. And then when we map out the response, the fractional change in fluorescence as a function of the direction that the bar moves, a subset of those cells are going to be direction selective. So this is a cell that prefers motion that's upward, and you can see a transient at the leading edge and trailing edge of that bar when the bar moves up, and when the bar moves down, we see a very small calcium response. Okay, so this is the tuning curve of the cell, and we represent the tuning curve of that cell by looking at the delta F over F amplitude as a function of direction, and then we get the vector sum. And that arrow is pointing in the preferred direction of the cell, and the length of that arrow tells you how strongly tuned the cell is. Okay, and so when we do this for a lot of cells, just give you a sense of how many cells there are, The green here are all the cells with the calcium dye. The blue cells are all the direction selective ganglion cells that we fall in this field of view. And luckily, we have some transgenic mouse lines that express GFP in subsets of direction selective ganglion cells. And this HB9 prefers cells that prefer downward motion. Okay, so that's going to be a groundish, truthish that we have that we're going to use later. And so when we do this now over the retina, we can see all 7 types. So for the on-off, so these are, they're called rose plots. They're polar histograms of the preferred directions of the cells that we find. And you can see for the on-off cells, we have 4, they cluster in 4 different directions. And in the on direction selective ganglion cells, we have the 3 different axes. And if you note, so things that we found using this, mapping out to describe this map, is that they actually have similar preferred directions, the on-off and on direction selective ganglion cells. There is this overrepresentation of nasal preferring direction selective ganglion cells for the on-offs, and there's an underrepresentation for ons, and that's just kind of interesting because that's the direction that represents optic flow, which is actually the dominant visual stimulus for these mice, and that we see this skewing of the maps. Do I have a little? Yeah, little arrows that you can see. If you're nasal or temporal retina, you kind of have these arrows pointing towards the singularity. And so I really want to emphasize the singularity because when we're thinking about developmental mechanisms, that to me looks like they might follow a molecular gradient. And that's actually one of the things that we're interested in pursuing. So, okay. All right, so now I've characterized what the map looks like. So now I want to get at this question on what breaks the symmetry. So like, what do we know about this direction selective circuit? And we understand this pretty well. So we know that this asymmetry is mediated by inhibition. And we know this for a variety of reasons. You could block GABA A receptors and your direction selective ganglion cell will respond equally to bars moving in all directions. So it loses its directional preference. We also know that if we do a voltage clamp recording and look at the synaptic inputs onto a direction selective ganglion cell, when the bar of light moves in the null direction, we have very strong inhibition. And when the bar of light moves in the preferred direction, we have very little inhibition and roughly equal excitation. And so you can think when the bar moves in the preferred direction, excitatory inputs come in, cell fires action potentials, bar of light moves in the null direction, you have excitatory inputs, but a huge amount of inhibition, and that prevents the cell from spiking. And so this null, the lack of response in the null direction is because there's a lot of inhibition. So we know it's due to inhibition. And we actually know the cell that provides that inhibition. It's this absolutely beautiful cell that's called the starburst amacrine cell. And we know this is the source of GABA for these cells. And we know that for a lot of reasons. If you kill starburst amacrine cells, the animal loses its ability, it loses direction selectivity. And we did an experiment in the lab where if you pharmacogenetically silence or actually slightly hyperpolarize these starburst cells, the ganglion cells around it, direction selectivity goes away, and then the drug washes out and direction selectivity comes back. So we know that you can even just hyperpolarize these cells a little bit and you get rid of the computation. So
horizontal do nothing. Horizontal inhibitory.
Horizontal cells?
Yeah.
So I would say they do nothing for this computation. I mean, it's always hard to say they do nothing. I'm sure they do something, but yes. Yeah, I did just say nothing, but it's always you can't say nothing. I'm sure they contribute to it. They're very important for contrast enhancement, but I will say, I'm going back to they do nothing because we can change the contrast and we can even blur the edges and we still get this direction selective response. And so I think what the horizontal cells do is not helping this computation, this particular feature. But starburst amacrine cells super symmetric, right? So like I still haven't told you how the symmetry is broken. And it turns out the symmetry is broken. The direction selective computation happens in the process of a starburst cell. And I'm going to show you the evidence for that. So if you look at one process of a starburst cell, this is a starburst cell that's filled with a calcium dye. And the thing to know about direction selective, about starburst cells is they have glutamate, they're a weird cell in that process is both an axon and a dendrite. So it's got glutamate receptors in kind of this part here in the inner 2/3 of it. And it's got release sites out in the outer part. So these kind of bright spots are release sites. And if you take a bar of light and you move it in the one direction, you get a very big calcium transient out at this varicosity site. And if you take that bar of light, whoa, and move it, oh, can I not see the other direction? Bar of light out, can you do it? Bar of light, you get a very small calcium transient. So, and if you do this across the whole starburst cell, right, you can look and see that the preferred direction of each of those processes is for outward versus inward. So there is this directional computation, but it's incredibly symmetric across the cell, right? But I want to be super clear, that is where the computation happens. That is the only place that cares whether the bar of light moves left or right is in one of these particular processes. And so then, so we know that more GABA comes out when the bar of light moves that way versus this way. Bigger calcium transient, more GABA comes out. And so you need a second thing to give you direction selectivity, and that is an asymmetry in wiring. So it turns out this is now a starburst cell that's located on the null side of direction selective ganglion cell, and it forms more synapses than a starburst cell that's located on the other side. In other words, if this process will preferentially form synapses with the direction selective ganglion cell where that's its null side. Right, an incredibly specific wiring that happens. Okay, so that is crazy, and I'm going to show you why it's crazy, because it turns out there's a ton of starburst amacrine cells. So these are all the starburst amacrine cells that are in the retina, and if you look at them, they have a huge, thick over-arborization of starburst processes, and those are the dendrites of a direction selective ganglion cell. Yet, there still is some incredible specificity that this process of a starburst cell will only form synaptic connections with a direction selective ganglion cell that it's its null side. Okay, and that is, and so is there evidence for this incredible asymmetric wiring? And there's two bits of evidence I'll show you here. One is there was a serial EM reconstruction that was done by Kevin Brigman, Moritz Helmstetter, and Winfried Dank, where they did calcium imaging in a retina. They found all the direction selective ganglion cells, and here are the 4 subtypes that they found that are shown here. And then they reconstruct those direction selective ganglion cells, and then they reconstruct the starburst amacrine cells that provide the inputs to them. And what they found is summarized here. This is now from the perspective of a starburst amacrine cell, soma, that this process, the red process, say, forms synaptic connections with that direction selective ganglion cell, while this purple process forms synaptic connections with that direction selective ganglion cell. Even though there's a red one sitting there, it doesn't form many connections with it, right? So there's evidence at the EM level. And then in some work that was done by our lab, we did it with paired recordings. So if you take a starburst cell that's located on the null side of a direction selective ganglion cell, so that this process is overlapping with the dendrites of that cell, you get a strong GABAergic conductance. If it's located on the preferred side, you get a weak GABAergic conductance. Okay, so functionally there's an asymmetry, and at the EM level there appears to be this asymmetry, right? And so that's it. That's the components that you need. So here's our starburst amacrine cell. It has this directional preference computation that's done in its process, but the only way that that can convey direction selectivity to the ganglion cell is if there is this incredibly precise wiring. So you need 2 things to give you direction selectivity, the computation in the starburst amacrine cells, and then the asymmetry of the wiring. Okay, so that is our circuit, and that is what and remember, now this has to happen essentially 7 times, right? There's 4 direction selective, there's 4 cells shown here, and then there's these 3 other on direction selective ganglion cells. Okay, so that's the question in our lab. What instructs this? How does this arise during development? So the first thing I'll say is that it's there right when the eyes open in a mouse. So a mouse is born with its eyes closed, its eyes open after 2 weeks. And as soon as their eyes open, you can, and you do one of these calcium imaging experiments, you can see pretty good, it's not perfect, but you can see very nice direction selective ganglion cells right when the eyes open. You can even see it when you image a couple days before eye opening. We also know by doing paired recordings that this asymmetric wiring actually appears between postnatal day 9 and postnatal day 11. And we could actually do, when we did those paired recordings, these were experiments done by Ryan Morey while he was a graduate student in the lab. We know that and actually Wei, well, she was a postdoc in the lab. Wei now has her own lab at University of Chicago, where she continues to work on direction selectivity. And so they were able to show that asymmetric wiring appears between postnatal day 9 and 11, right? And actually, I'll show you this there. So here's those paired recordings. If you do it at postnatal day 4, whether the starburst cells on the null side or the preferred side, they're weakly connected, but all the starburst cells are symmetrically connected. Postnatal day 7, that's true. Postnatal day 14, you have an asymmetry that's as big as that you see in adulthood. And then Ryan went on and did this to really narrow it down to around postnatal day 9, things are symmetric. Postnatal day 11, they're asymmetric. So it's really a 2-day period. Ryan, in his experiments, did a quantal analysis and was able to show that the change that happens in that 2 days is number of synapses instead of strength of synapses. So there's a burst of synaptogenesis that's happening in that 2-day period, and it's only happening between the correct process and the correct postsynaptic cell. So we think it's due to new synapse formation. And so that seems to be, and then we also, so the asymmetry is there, and we wanted to see whether or not the starburst processes are direction selective. This is very new data from a new graduate student in the lab, Maya. And when you do an expanding sort of bullseye of light, you get a strong response, and you have a contracting, and you see a small response, and that is there right at eye opening. So the data that you're looking at here is actually in a P13 mouse. So the direction selective process also seems to be there. So everything seems to be there right when the eyes open. Okay, so what's happening between P9 and P11? So if you look at the morphology of a direction selective ganglion cell, dendrites, nothing. So they look stratified. They're stratified very early. They're morphologically mature. Nothing seems to change during this time, but the starburst cell changes a lot. So here you're looking at a starburst cell that is during postnatal development, and you actually see it goes through a lot of changes at this time. At postnatal day 10, the process is filled with lots of little filopodia, and those filopodia between postnatal day 10 and postnatal day 14 actually seem to disappear, and the varicosities appear. And this is work that was done by an undergraduate in the lab, Xia Xu. And so here, if you look at one of these processes at postnatal day 7, by postnatal day 14, you see those varicosities appear. And again, during that same developmental period, the number of varicosities increases and the number of filopodia disappears. Okay, so this really is consistent with the synaptogenesis that's happening. Yeah.
For the synaptogenesis, is that at the glutamatergic sites or the GABAergic sites?
These are at the GABAergic sites.
And
yeah.
how mature is GABA in like a P9 rat?
And so by mature, you mean is it depolarizing or hyperpolarizing? So at that point, it's hyperpolarizing. So P9 is hyperpolarizing. It looks like when that switch happens might be postnatal day 6 or 7. Yeah. Okay.
I have a question. If it's like at 7 directions,
Yeah.
what happens that it
Uh,
like you're more
Yeah, all 7. We can find all 7 right when the eyes open. So
maybe I'm just missing something. So there are 7 types of like 7 directions.
yeah, so I'm showing you just the 4 to make of the on-offs that are shown here. There's these on direction selective ganglion cells, which are the ones that drive this reflexive eye movement. And there seem to be 3 of them that have roughly the same preferred directions as 3 of these cells here. But they're a different cell, right? They have a bigger dendrite. They send their axon to a different part of the brain, but they get inputs from starburst cells. All of them lose their direction selectivity when you hyperpolarize the direction selective, the starburst cell. And they all seem to get their inputs from starburst cells. So same kind of retinal computation. But yeah, is that answering that question?
I guess what I'm asking is that I'm expecting to see 8.
Yeah. Okay. So
what happens?
what happened to the other one? So there was this overrepresentation of nasal in the on-off ones and an underrepresentation in the on. Okay, so there's actually literally a debate in the field. There's a world in which we talk about this. If we lower our threshold for what we call a direction selective ganglion cell, we will see one of those on cells seems to become, we can now see responses. in that direction.
Then from an evolutionary perspective, they're better at detecting nasal.
But they're not as tuned. They're not as strongly tuned. So so they, again, these cells are very highly sensitive to the whole visual world moving. It's whether or not they have a stronger response to one versus the other. So there are ones that just have a more symmetric response to left versus right. And so for this nasal, but they're there and they're very responsive to that stimulus. They're just not as selective to that motion. So, right, because clearly you have an eye movement in that, reflexive eye movement in that direction. And so now it just becomes, what's your criteria for being direction selective? What's your threshold? And so we find fewer of them cross that threshold.
So are you implying that connections are made specifically right from the onset? Or are you open to the possibility that there's an addition of connections, but that can be coupled with some removal?
Yeah, sure. I'm open to that.
Okay.
I'm open to anything. Okay. So, so we know when it happens. When you're studying development process, that's the first thing you need to know is when. So we know P9 to P11, and now we've been trying to break it. So we've been trying to do a lot of things to break direction selectivity. One of the things we did is we used a mutant mouse that we got from Alex Kolotkin's lab that was a SEMA6A mutant. It really messes up the morphology of the starburst cells. They get very, they get smaller and they get distorted, but we still found that each process was still direction selective, and when it crossed the dendrite of the right postsynaptic cell, it still formed the synapse with the correct one. So it did not seem to break any of the rules that we had. So it doesn't seem to depend necessarily on the shape of the starburst amacrine cell. And some experiments that were done by Marla Elkesny while she was a graduate student in the lab, she actually did something where she focused on this one direction selective ganglion cell that has an asymmetric dendrite, because that's the one that you might argue there's an asymmetry postsynaptically, and that would, might facilitate this contribution. She found that by dark rearing animals, you could actually flip the orientation of that direction selective the orientation of the dendrites of that cell, and that had no effect on its direction selectivity. So the wiring cues were still there. So it didn't seem to depend on essentially the amount necessarily, anyway, the orientation, any asymmetry in the postsynaptic cell. That's what we concluded from that. Okay, so, but of course, so now we're gonna tell you the two things we're doing now to try to break it. We have found one thing that breaks it, but the first thing, maybe particularly those of you who study development, is I've drawn these colors, like blue and yellow and purple, and it makes you think there must be some molecule, right? There must be some molecular code that, bizarrely will be differentially transferred or, localized in the different processes of the starburst cell that recognize some other molecular code on the direction selective ganglion. So that's got to be part of it. And so we wanted to have some search to figure out what that was. And so, and this is work that was is being carried out by Josh Twarig, and to get at this question. So we did a bulk RNA-seq experiment, which was based on this idea. We had mouse lines that fluorescently label rightward preferring direction selective ganglion cells. We actually have two mouse lines that prefer right. And then we have this one mouse line that prefers down. And we took P10 mice right at the period of time when the sorting has happened. We dissociated the cells of the retina. We did FACS sorting, and we looked for things that were differentially expressed, that were similarly expressed in the two rightward preferring DS cells and different from the HB9s. He enriched for molecules that are known, or genes that are known to be involved in synaptogenesis and cell adhesion. And we got down some really the biggest candidates. And the biggest candidate that Josh decided to pursue is a molecule called cerebellin-4. And cerebellin-4 has been implicated in inhibitory synapse formation. And in fact, it was implicated in cortex for targeting of particular inhibitory interneurons to different parts of the pyramidal cell process. So we really liked it as a candidate. We did some control experiments to show that its expression was much, much higher in the ventral preferring cells over the rightward preferring cells. And that it was strongly expressed in those, we did in situs, which is a really new thing for our lab, so this is a major accomplishment. So we did in situs that showed more strongly expressed in these ventral preferring cells versus the nasal cells. Okay, so that'll look good. And so then we got a mouse that Tom Sudoff had developed, which was to knock out, that's essentially a flox cerebellum 4. And where there's a green reporter and all the cells that have cerebellum 4, and then if you cross it with a Cree line, where we knocked it out of all ganglion cells, the cells where it gets knocked out of turn purple. And so, so just to give you a sense that these are all the cells that are cerebellum 4 positive. You can see better here. These are all the cerebellum 4 positive cells, and this is an example of what the knockout looks like. There's a lot of purple cells in the background. Unfortunately, cerebellum 4 is also expressed in a population of amacrine cells, not involved in DS, but it's there. But we can knock it out of ganglion cells. Okay, so now we've got our mouse. And so the first thing we did was to do our calcium imaging to see whether or not there's a disruption in direction selectivity. And we crossed it into these ventral preferring cells, and we found that indeed there was a small-ish effect. So if you look at, these are the fluorescence traces that show you the different preferred directions. And this is these guys prefer ventral, over dorsal. So there's a little bit of direction selectivity, and that is slightly reduced, but still apparent in these knockout mice. And so the phenotype, this is a cumulative histogram to show you there's a slight leftward shift in directional tuning, but basically they're still tuned. We wanted to look at the synaptic level at these cells, and so Josh did voltage clamp recordings from these cells. He finds that, again, in a control, you have strong inhibition for bars moving in the dorsal direction and less for bars moving in the ventral direction. And then when he did this in a knockout mouse, he found that there was overall a reduction in inhibitory synapse formation, but it was still asymmetric. And so cerebellum 4, so again, this is looking at the peak inhibitory amplitude, and in controls, if it goes in, actually this is just, it's stronger and on versus off, but just look to the right here. This is the direction selectivity, and you can see that there is they're just as direction selective in the knockouts as they are in the wild type, although there is some variance, but they're overall a reduction in the amount of inhibitory synapse formation. Okay, and that excitation was unaffected. So from this, we think that this showed that cerebellum 4 plays a role in generating inhibitory synapse formation in general, and that reducing inhibition overall will reduce the amount of direction selectivity, but this was not the magic bullet, right? And this is something that, it was a bummer. This is something that's, and it's just kind of a 4 years of work, small phenotype. This is why I don't like RNA-seq. We have to get better at doing our analysis so we can pick a better candidate. And I'll tell you right at the end of a new strategy that we're using in order to do that. Okay. In addition to synaptogenic factors, we're really interested in this idea of molecular gradients, right? And I told you that there is this ventral, this sort of pointing towards the ventral pole for at least those ventral preferring cells. And so we looked for molecular gradients that exist in the retina, and it turns out that there is one that emerges from the ventral pole, which is one for the ephrin Bs, the F Bs and ephrin Bs. And I think maybe, or if you know, that ephrin gradients have mostly been implicated in retinotopic development. So what we're looking at here is kind of the retinal gradient versus the gradient in the superior colliculus, and that ephrin A's were important for sort of horizontal map formation, ephrin B's, and for vertical map formation, retinotopic map formation. And it turns out the FBs, the FAs seem to have this linear gradient across the whole retina, but the FBs, we explored a little bit more, and it looks like if you look at, and we have a triple mutant in that mouse. And the first thing we did is we looked at the receptor expression in the retina, and it's a little bit hard to see from here, but when we do a receptor fusion protein with Dave Feldheim's lab at UC Santa Cruz, we actually do find it emerges from a ventral pole, right where all those DS cells are pointing. we got this triple mutant mouse, however, and unfortunately, this is very preliminary, but we still see this ventral pointing towards that. So there's a lot of other gradients in the retina. Anyone who has ideas for other gradients that we might want to look like on the dorsal ventral axis, but this was the one that we knew about and we had worked with before, and that didn't seem to affect at least this pointing. But that's another area that we're pursuing. Okay, molecular code didn't work so well. But we finally, and it didn't break it. We still haven't broken it. We finally broke it. And this is what I'm talking about, some work that was published just last year about what instructs the development of direction selectivity. And the beauty of all of this is what seemed to break it is blocking retinal waves. And this was, Reese alluded to this at the beginning, but let me just introduce what retinal waves are. And this is work I'm going to talk about done by Alex, Maya, and Karina. Okay, so retinal waves are an activity pattern that is present in mice prior to maturation of vision. So as I was saying before, mice are born with their eyes closed, and importantly, the photoreceptors at this stage of development are not synaptically connected to the ganglion cells. So before, at around postnatal day 11 is when those synapses form, but before that, there's no light response in the retina. So there's no photoreceptor generated light response in the retina. But the retinal ganglion cells are not silent, they're spontaneously active. And that spontaneous activity we call retinal waves. To give you a sense of when this is in development, I just wanted to throw this schematic of human brain development before birth. So the retina, or the eye, develops independently of the brain. And the axons that the retinal ganglion cell axons, the optic nerve forms incredibly early. And just in this period of development we're talking about is about 5 months in humans. So mice are born at what's around a little bit before 5 months in human development. Not a lot of visual experience, but there is this spontaneous activity. So I just want to show you this new movie that we took with this cool macroscope. So this is a microscope that is made, that is based on an epifluorescent scope with this very, very nice camera that has very small pixels. And we did some great engineering done with Ben Smith so that we have, the bottom line is we have a field of view that's 5 millimeters across with 1.5 micron resolution. And we use this to measure retinal waves to give you a sense on how beautiful this macroscope is. This is an image of the entire retina, mouse retina. This is a mouse retina that has been labeled, has got GFP in a particular ganglion cell type, the IPRGCs. So you can just see the kind of cellular resolution we can get with this microscope while looking at the whole retina. And we use this to do calcium imaging to look at this spontaneous activity. And so what you're looking at here is the spontaneous retinal activity that you can see in this very immature retina. And that's why we call this retinal waves. This particular movie is playing at about 5x real time. I'm now going to show you in real time what's happening in that square. And you can see those are all ganglion cells that are sort of depolarizing by these waves that are propagating across the retina. Okay, so this is a robust source of activity. It activates starburst amacrine cells. It activates direction selective ganglion cells. And so we looked to see whether or not it was involved in setting up direction selectivity. And so so we know that as retinal waves are present for an extended period during development. During that period of development, the retina is maturing a lot. And so there are different circuits that mediate waves. This cholinergic circuit happens in the first postnatal week. That's when the starburst amacrine cells and the ganglion cells are getting co-activated by these waves through a cholinergic circuit. And later on, you have a glutamatergic circuit. Remember, development of direction selectivity is happening during these glutamatergic waves. But we have a mouse that doesn't have waves in the first postnatal week. Okay, so we're going to disrupt activity here. Synapse formation happens here, and that's the thing that breaks the direction selectivity. And this is to show you how it breaks it. It's really interesting. So this is in the adult, as we showed you before, at eye opening, direction selectivity is there. In this mouse that doesn't have waves in the first postnatal week, we have no horizontal direction selectivity. But we still have vertical direction selectivity, but we have no horizontal direction selectivity. And in fact, we know that this mouse that is lacking these horizontal waves actually lack an optokinetic reflex in the horizontal direction and has a normal one in the vertical direction. So something about these retinal waves is blocking horizontal, but it's not blocking vertical. crazy, right? But thank God it broke something. Okay, so whoops. I seem to have, so how do waves influence development of horizontal direction selectivity? Well, the first thing that Karina checked was to make sure the cells hadn't all died. And so we actually have a GFP marker in those cells. And when you do imaging from those cells, they're normally direction selective. But in this mouse that doesn't have waves in the first postnatal week, they are still responsive to light. They're still there. There's just as many of them. They look the same. They just have a symmetric response to light. She's now done voltage clamp experiments from those cells, and she finds that they have a more symmetric inhibitory current. So if you look at the amount of inhibition in a control in the null direction versus preferred, there's a big asymmetry. They're not totally flat, but there's less of an asymmetry. So there's a little bit less of an asymmetry in inhibition. So we know that, but the question is now why? So Maya has been looking at the direction selectivity of the Starburst processes. The first thing she actually wanted to do is to show that Starburst cells participate in waves. And so this is just an example of calcium imaging from a single Starburst cell to see what their participation in waves look like. And I think the key thing to note here is that every wave activates the entire Starburst cell, and there's no branch-specific activity at all. The whole thing seems to go. And there seems to be no spontaneous activity in between the waves. So the only thing that activates the Starburst cell seems to be the waves. So that is one thing she found. And then when she repeated her experiment of this expanding versus contracting bars, she found, again, there was an asymmetry. Expanding bars gave a bigger calcium transient than contracting, but that asymmetry was less. So it could be, and we're currently doing experiments to check on if the wiring has been disrupted in some way. So it could be horizontal has been broken because of it's a death by 1,000 cuts, or we haven't found out yet what the waves are doing to break the circuit. Then so we can begin to get at the mechanism that of what they might be, what at the molecular cellular level is actually being broken. That affects horizontal differently than vertical. So there you go. Lots of unpublished stuff, lots of mysteries. But what we do know is the following, that DS circuits emerge during this 2-day period, during a period of robust synaptogenesis in a manner, at least for the horizontal cells, that depends on retinal waves. Vertical does not. We're trying to identify the molecular candidates. Our bulk RNA-seq gave us cerebellin-4. Appears not to be the primary determinant of vertical DS, but we have other candidates in the pipeline. We now have this mouse that doesn't have horizontal direction selectivity, and there's a lot of information now at the single cell RNA-seq level that didn't exist before. And so we're collaborating with Karthik Shakar, who's a new professor at Berkeley and came from the Josh Sains effort, who's done a lot of characterization of retinal ganglion cells across development. We're now going to compare each of the 7 subtypes of direction selective ganglion cells, in the beta-2 knockout and in the wild type, compare it with our bulk RNA-seq. We're going to have a lot of RNA-seq data and people who know how to analyze that to hopefully come up with a candidate that gives us a better result than cerebellin-4. And we're continuing to pursue molecular gradients, which I think is important, will not be revealed by RNA-seq, right? So that is not likely to be something that we, but might be the thing that gets disrupted in the absence of waves. We know horizontal direction selectivity requires retinal waves, so that seems to be true. We know it's probably due to the breakdown of this asymmetry in inhibition, but there's a lot of ongoing experiments now to figure out whether it's the direction selectivity of the Starburst process or the wiring that is really causing that asymmetry. All right, so here are the people who did the work. We love retinal waves. Got it, sorry. And so this is the lab. Actually, a lot of these people are no longer in the lab. Christian Buffo just graduated, and Josh Twerk also just graduated. So so these are the and Josh is going to stay in the lab a little bit longer. So I just want to make a note, because I know there's some graduate students here, just to tell you sort of where the last few graduate students from my lab have gone to. So Franklin Cavall Holm is actually in a retina lab doing a postdoc at Harvard. He's in Michael Doe's lab. Christiane actually just accepted an internship at the World Bank, where she's going to be working on education programs in STEM education in sub-Saharan Africa. Matthew, who has also worked on direction selectivity, is now at the Allen Brain Institute working on the Connectome project there. Malik El-Kesni is now at the Institute for Photonics in Barcelona working on some totally different organoid sort of project. And Alex, who I did mention, is now a professor at Vanderbilt. And I kind of want to say that because your PhD does not lock you into anything. There's a lot of different things you can do after you get your PhDs, and so I just wanted to sort of make note that that it opens lots of doors what this sort of research can do. And all my research is funded by the National Eye Institute. So that's it. Thank you. All right. Anyone have ideas? Pete.
So, I mean, one thing that I guess scares me a little bit is, like, even with the beta 2 knockout that completely wipes out the retinal waves?
Not completely. No.
Okay.
Changes them.
Because it doesn't, you don't completely lose the horizontal selectivity.
Right.
So it could be, and it almost certainly is, multiple interacting things. And so, like, truly knocking these things out might be a really challenging thing.
Yeah.
So what are your thoughts on that? And then the other thing is, do the waves influence like Ephrin expression? Is there any evidence for that kind of thing?
Yeah, so I can answer the second one first. So that's been looked at by David Feldheim's lab, and they do not. So the waves don't seem to affect Ephrin A expression, but people haven't really looked at Ephrin B. And it's interesting, you know, this horizontal versus vertical is kind of an, I don't know if this is relevant or not, but when people, so we've known that retinal waves are important for retinotopic map formation. So if you disrupt in this beta 2 knockout, the one axis of the retinotopic map gets disrupted. But it turns out the other axis, which is encoded by Ephrin B, so nasal temporal gets disrupted in the retinotopic map, but dorsal ventral does not, right? So that was already known, like horizontal is more sensitive to retinal waves than vertical for retinotopic map formation. Maybe Ephrin A's or Ephrin B gradients are more sensitive to that activity or the readout of that activity. So the gradients themselves don't seem to be sensitive to activity levels, but it doesn't mean what reads out those gradients isn't sensitive to activity levels. And that's kind of what the thought might be there. So the question is, how can waves, so waves seem to go in all directions. It turns out, so how could they instruct, how could they break symmetry in any way? So it turns out there are more waves that go nasally on the retina that encode for optic flow. There's a bias towards that, but that bias persists in the waves that, so beta 2 knockout waves are different in that they're very big and very fast. They're kind of almost like flashes, and they're very infrequent. And so, but they have the same, actually, I take all this back. There are very few waves that happen there, but it's the propagation bias here that we think matters. It's in like the glutamatergic wave. So beta 2 knockout mice have glutamatergic waves. And so the propagation bias of those waves, which are the ones we think would matter because that's when the synaptogenesis is happening, that's not different. So the infrequent waves that are there in the beta 2 knockout, we can't really measure their propagation bias because they're flashes kind of across the whole retina. So it's just weird. The manipulation there is affecting something that happens here. And at least the propagation bias isn't different in the beta 2 knockout. So I am doubtful that the waves are instructive for this map in any way. And it could be they're reading out the same thing that's setting up the DS map. The DS map has some GABAergic gradient, maybe the same thing is affecting both propagation bias and DS circuits as opposed to it being, I don't know. Yeah.
That was a great talk, thanks. I was wondering if in this beta 2 knockout, if eliminating or at least disrupting these waves during the first postnatal week is affecting something else besides DS, like, I don't know, like other ganglion cells or downstream wiring, upstream
Yeah.
wiring, et cetera?
Yeah, so we, our lab and many labs have shown this is a very robust disruptor of eye-specific segregation in retinogeniculate projections and of these retinotopic maps in the colliculus, and a variety of other things. So people have found quite a variety of things that have been disrupted in cortex in these mice. They don't have depth perception. There's alignment of a couple of other maps in visual cortex that get disrupted. So they can see, they're just a little bit blurry in what they can see. So their acuity has gone down, but they can still see. But they seem to affect the organization of retinal projections and then maybe within visual cortex. Within the retina, I have spent the last 10 or 15 years looking for phenotypes in the retina, and this DS one is the first one that we found. Doesn't mean there aren't others, but it's the first one that we found. So we found something in these IPRGCs too, but that's a little more, it's a smaller phenotype than what we see in for direction selective circuits. So
Do
hopefully
you
we'll
think
find
there's
other
any
things.
like feedback or anything like that?
From the so okay, the answer to that used to be there is no projection back to the retina from the CNS until 2 years ago, and it turns out there is a projection. So in birds, it was known. It was not thought to be present in mammals, but it turns out there is a histamine projection that now comes back, and it's actually pretty interesting. Actually, Michal Rivlin, who is a postdoc in my lab, has described it, and it's only in either the ventral half or the dorsal half of the retina, but it's like a histamine projecting neuron into the retina that seems to target histamine receptors on some amacrine cells, but it's only in one half of the retina, and I can't remember which one.
Okay, so it's not on the DS cells.
It doesn't seem to be on the DS cells. Yeah. There is another weird thing that happens, which is transiently during development. So, retinal ganglion cell axons go to the optic chiasm, and they either choose to stay ipsilateral or contralateral. There's a small percentage of them that go to the other eye, right? So they make a mistake and they grow back to the other eye. That's a transient thing that seems to be gone by around postnatal day 4 or 5, but that's something I suppose we can't ignore, one eye influencing the other. But I'm just throwing everything out there I know in case it's important. Questions? In Zoom world, are there questions out there? Is there a Zoom world?
Someone
Yeah.
said that area.
Yeah.
I was just trying to decide whether this is a good question or not, but I'll ask anyway.
It's really good to ask any question. It inspires all the young people.
So it seems really convenient for the mouse, just from an ethological perspective, that the direction selectivity is organized in this way, right?
Yeah.
There's a sort of a horizontal and a vertical, and you talked about the animal's own movement and the optic flow and all of that seems to match. Does that emerge just from the anatomical structure of the retina itself? Because it's all of these things are there before the eye opens, so it's presumably somehow emerges. But my understanding of the retinal waves is that they're kind of going in all different directions.
Yeah.
So I can't quite get my head around how these two things interact.
Yeah, so I call these things non-answers. I have a non-answer for that, but it's something which is, so not all animals, not all vertebrates have retinal waves. And the ones that don't have retinal waves are like Xenopus, and zebrafish actually have retinal waves, but they also see very early. But let's talk about Xenopus. Xenopus don't have retinal waves, and they swim at this stage of development. So they have a lot of optic flow at the same time that these retinotopic maps are forming and when the direction selectivity maps are forming. And so the thought is that, so for lower vertebrates, they have optic flow. For mammals, they don't, because they just sit there, right? And so it could be that these waves were this evolutionary replacement for what normal optic flow would be. It's a non-answer because it's like it's a rational way to think about it. But there were these very cool experiments done by Holly Klein where she raised Xenopus so that they had reverse optic flow, and it greatly disrupted the retinotopic maps, only horizontally and not vertically. And so it does, again, seem like there is this horizontal optic flow signature motion nasally on the retina that's key, at least for retinotopic map formation. So I don't know if that's why, but the how and like what breaks the symmetry in this immature retina to give you a propagation bias or something else we're trying to understand. So
Thank you.
it's very mysterious. She's just putting on her sweatshirt.
So the pattern of waves, is
Yeah.
it random?
Yeah, so it's funny. We have spent really my career studying these things. Pete, am I right? So I've been my career. Pete studies we started working on this around the same time when I was at the NIH. Looking at and Pete studies I like to call them the retinal waves of the spinal cord, which Pete hates it when I say that. So, but he sees waves.
These are spinal waves. Right?
Yeah, or spinal waves in the retina, however you want to think about it. And so this question I have been thinking about for a long time. And now we have this macroscope, right? And so this macroscope allows us to really look to characterize those properties. And so we were looking for these asymmetries. And right now, one of the things that Maya is trying to figure out is where does it arise? And does it arise locally? Like no matter where the wave starts, does it always go kind of nasal? Or what it looks like she's finding is there's an initiation bias that more of them start temporally. So when you're just imaging in nasal retina, it looks like more of them are going nasally. But if they're isotropic and more of them start here, you're going to see a bias that is in that direction. So she's in the middle of doing that analysis now, because that's a very different thing. It could be it's isotropic and it's just where the initiation is. What makes a wave initiate? There's some ion channel, and maybe there's some developmental gradient of that ion channel that that leads to that spontaneous depolarization. Versus there's some asymmetry very locally within the retina that is always pushing waves to go in one direction. And I think once we know that answer, then I think we can look for what that might be. But they seem to be, so if I had answered that question a year, or how long we had it, 6 months ago, I would have said, yes, their initiations are random and that they propagate randomly. But now that we have this,
Nice.
now it seems like.
And you mentioned spinal waves. I know that's outside the scope of this talk, but
Yeah.
what are those about? Are they also?
So this spontaneous activity is actually found in a lot of parts of the developing nervous system. So it's in the developing cochlea, in the ear before the hair cells mature. There's this spontaneous activity that propagates along the cochlea. That's important for setting up sort of the auditory maps. In the spinal cord, there's spontaneous activity that the motor neurons participate in. That is actually mediated in part by a cholinergic circuit. That's a transient circuit that's there. And that is important for motor neuron, I don't know if it's navigation or innervation or maybe even driving neuromuscular junction refinement, right? So these are all, but this is what Professor Wenner's lab studies.
Synaptic strength and intrinsic excitability as well.
Yeah, right. They've done beautiful work to talk about how it's very important for synaptic strength within the spinal cord itself. So they're farther ahead in the local circuit than we are.
I guess hippocampal.
And in hippocampus, they're absolutely there. There, it's much more governed by this fact that early in development, GABA is depolarizing. And so in cortex and in the hippocampus, there are these waves that have been studied, but they really go away when GABA becomes hyperpolarizing. So there you just have this, but they also have, there's work from Rosa Casart's lab that has shown that they actually have nuclei. There's some parts of the hippocampus where they're more likely to initiate, where many of the waves initiate from. So that's different. It's in the locus ceruleus. It's in lots of different parts. Yes.
Just a really quick question. During development, during the 2 days, before that, how does exposure to light affect the retina?
Yeah, so we have dark reared and not found anything earlier on. So we don't seem to think that that's important, particularly for this circuit. But it turns out there are some light sensitive cells. These are these intrinsically photosensitive ganglion cells. Maybe it's important for myopia or the prevention of myopia. There's lots of things that that we haven't looked at that it might be important for. And so there's actually really interesting work that shows even embryonic development in mice that dark rearing prevents the maturation of the vasculature. And so this is like light that gets through the uterine wall in a mouse that activates these iPRGCs, was very important. And these iPRGCs are thought to release a growth factor that's important for vascular development. So there is some stuff that seems to go on earlier, so we haven't really looked. But for the DS circuit, it doesn't seem to matter. All right. Thank you all.